Hi, I'm Brian Carlson, and I'm going to show you how to annotate models using SemGen. So the first thing I have to show you is where to get SemGen. And SemGen can be found at this Semantics of Biological Processes website. You can see the URL here. You will link on this cross-platform release 64-bit link, and you will download SemGen 3.0 beta, which I might add requires Java 1.7 or higher, 64-bit. This version is not going to be fully functional on Mac machines, but is fully functional on the Windows machines. Just a caveat right there. So I did indicate that you need Java 1.7 or 1.8, so in order to find out whether you have Java 1.7 or 1.8 installed, here's a website that you can go to, but I can show you really quickly on this machine. You go to Settings, this is a Windows 8 machine, Control Panel, and you go under Programs, Programs and Features, and you will see the Java 8 update 64-bit. If you don't see that, if you see something that's not 7 or 8 and not 64-bit, I recommend you uninstall and then reinstall a Java 7 or 8 64-bit from the Java website. So that out of the way, we want to get a model now. So the CellML website is the place to get that, and this is the URL for the CellML website. Our friends down at the University of Auckland in New Zealand have compiled a list of computational models drawn from the literature. They've curated them to make sure that they run properly and reproduce figures if placed into a simulation environment. And so you can actually go through this. These are kind of ranged by categories. And the category we're going to look in is the excitation contraction coupling category. We're going to actually pick the model by Nieder, Hunter, and Smith. And so this is a typical model web page. It shows a model description, which is indicated here. It has a figure that shows the model structure and how things are connected. It has a sidebar where underneath downloads you see a couple different things. You can actually just download this file and you'll get just the CellML file that's associated with this model. So if you click on that, you will get something downloaded into your download folder. I've dragged both of those items onto the desktop here, so let's get started and see what we can do. So here's SemGen. We're going to start it up. And there's many things you can do in SemGen, but the first thing you want to do is annotate a model. So you click Annotate a Model. You select the Nieder, Hunter, Smith model. And this is what we get. So we get several different panes here. This pane has metadata that can be attached to the model, either drawn directly from CellML or you can actually input this yourself, but this is metadata for the whole model. These are the code words that are listed, drawn from the CellML model, and these are the submodels that contain these code words. This pane actually shows you the details, the annotation that is attached to the highlighted code word in the code word pane, and this is the raw CellML file here. And when you select a code word, it shows the first instance of that in the CellML file. So we're going to pick the calcium concentration of the cytosol here, and we can actually attach some free text to this, and we're going to call it cytosolic calcium concentration. Okay. And when we've done that, now you noticed F appears in this column here, which indicates there has been a free text definition appended to this code word. The other thing you might notice is several of these code words actually have a P in them. And this P means that SemGen was able to go through and, based on units, suggest a property. And in this case, for this CAI, the chemical concentration was selected by SemGen. If this was incorrect, we could actually change this, but it's correct, so we're going to move right on to attaching entities to this property. So let's add an entity, and this is going to be the chemical concentration of what? We select the binoculars here, and we know it's calcium, so we look at the different ontologies that are available here. The chemical entities of biological interest would be the one we want to pick, and we can type calcium star, the wildcard character, and we'll get a big list. And in this list, we'll see calcium ion, but we want something more specific. We want a calcium 2+. That's the specific ion that we're interested in. Now we want to actually say, where is this chemical concentration of calcium? And so we select here the foundational model of anatomy, and we type in cytosol. And here's a pre-coordinated term, the cytosol of the cardiac myocyte. That would work, but we eschew the pre-coordinated terms, and we actually like to compose the composite by not pre-coordinating. And so here's the portion of the cytosol that's going to be the proper entity to select here. And now we're going to add in, using again the foundational model of anatomy, myocyte, and cardiac myocyte. Apply. So that is the intended scope. If you read the paper, that it doesn't talk about whether it's a left ventricular, an atrial, or a right ventricular. So that would be the intended scope of the model, and that's where the annotation should end. If we wanted to save this model with all of the annotations in it, we select File, Save As, and we can type Nieder Hunter Smith 2006, and it'll be a .owl when we save it. So if we minimize this and look on the desktop now, we actually have a file that's a .owl file, and it actually contains that annotation we have just created. So this is the first installment of how to use SemGen. Stay tuned for future tutorials on how to use SemGen, and thanks for listening.